Hello viewers, welcome to my YouTube channel BioHub. I'm your facilitator Dr. Seema Sharma. Today we are going to explore one more very interesting topic from class 7 biology part, and it's from chapter number 1, nutrition in plants. And today we are going to explore the basic conditions which are necessary for photosynthesis. So let's explore So let's together explore this interesting subtopic, which is related to conditions for photosynthesis together. Okay, viewers. So we are starting up with the today's video. Viewers, because of this COVID pandemic, we are not able to come to school physically, whether we are teachers or students. And we have so many queries related to different subjects, not only biology, different subjects. So this is my small attempt for my students or viewers that they can stay home and they can learn about biology and various aspects of biology in a simple way with a simple explanation. And for that, today I'm again back with you people to explore about conditions for photosynthesis. Let's please join me and let's explore together this very interesting subtopic of class 7 biology part, chapter 1, nutrition in plants. In this chapter, if you are watching my videos regularly, then you must have come across my videos on this chapter part wise. I have already uploaded 2 videos of this chapter, and today it's my third video on nutrition in plants. And in this video, I'm going to cover conditions which are necessary for the process of photosynthesis. This is the journal layout of your chapter, whether you are studying from NCERT book or Living Science, or you are studying from any other publication. But this is the journal layout of this first chapter in almost all the science books. So let's together explore conditions for photosynthesis. But before that, if you are not watching, you have not seen my earlier videos, then, and for those who are watching regularly, just a small recap what we have explored so far. We are on chapter 1, nutrition in plants, and in previous video, that was part 2, we have covered two basic terms: autotrophic nutrition, which is carried out by all green plants, and that's why they are called self-dependent, and this mode is called autotrophic mode of nutrition. This autotrophic mode of nutrition is only possible because of one very important and interesting process, that is photosynthesis. So photosynthesis is a process, a stepwise procedure in which plants are utilizing inorganic substances and converting those into organic substance, that is sugar, to be more precise, it's glucose, with the help of sunlight, where in the chlorophyll containing parts of the plant. And mostly this photosynthesis, it is carried out by green leaves. So this is the equation of photosynthesis. Here the plant is using simple inorganic material like carbon dioxide, water in the presence of sunlight. In the chlorophyll containing parts of the plant, majority is leaf, and then it is converting it into glucose, that is C6H12O6, plus there is evolution of very important gas, that is O2, oxygen gas. So this is about the equation of photosynthesis. Now we know, viewers, we know has to carry out photosynthesis, it should have chlorophyll. And in plants only, this plant cell only, chlorophyll is present. Majority of the plant cells, they have chlorophyll and they are doing photosynthesis. So today in this video, what you are going to explore with me, that there are certain conditions for photosynthesis. And in addition to those conditions, we will not only learn about what are the various conditions for photosynthesis, we will also verify a few of those conditions with the help of certain experimental setups. So viewers, I'm starting up with today's video. It is on chapter number 1, Nutrition in Plants, and part 3. In this part, I'm going to cover conditions which are necessary for photosynthesis. Till now, my viewers my viewers know that for photosynthesis, presence of chlorophyll is must. Presence of light is must. There should be availability of carbon dioxide and availability of water. So these four things are necessary if the organism has to carry out photosynthesis. So the green plants, they are carrying out photosynthesis because their leaves, they have chlorophyll in them, and they are carrying out this process in the presence of sunlight, though this photosynthesis can be carried out by plants under artificial light, but the best sunlight is best for the green plants for the process of photosynthesis. There should be availability of carbon dioxide, and there is no dearth of carbon dioxide in our environment. You know, our activities, they are increasing the amount of carbon dioxide in the atmosphere, so it's available in ample. Availability of water, which the we are watering the plant, or uh, it's raining, that water is absorbed by the soil and then it is taken up by the roots and it is transported upward through xylem vessels. So these are the four conditions. So viewers, what we have to remember as far as conditions for photosynthesis are concerned, that presence of chlorophyll is must, presence of light is required, there should be a proper amount of carbon dioxide, there should be availability of water. If these four conditions are fulfilled, only then the plant will be able to do photosynthesis. For example, we are not watering the plant. The water plant will dry up. It will die. There will be no question for photosynthesis. If we are not providing carbon dioxide to the plant, we are keeping in such a manner that there is no carbon dioxide. The plant will not be able to do photosynthesis. It will not be able to make its food, that is glucose, and it will subsequently die. If we are keeping our plants in some dark area, then also gradually our plant will die. And if there is no chlorophyll, then there is no question for photosynthesis. So my viewers, please remember, four conditions are necessary for photosynthesis, and these are chlorophyll, light, carbon dioxide, and water. So let's proceed further. As I told you earlier that we will not going to learn about conditions for photosynthesis. We will also go ahead with the verification of conditions for photosynthesis. We will not say that, okay, it's written that presence of chlorophyll is must. We will also explore, is it must, is it required or not? And for that, I'm taking you a little further in the experimental zone. And here we are going to verify the conditions which are necessary for photosynthesis. We are starting up with first condition, that light is must for photosynthesis. So this is activity 1. We have to demonstrate with the help of one experimental setup that light is necessary for photosynthesis. So what we have to do, viewers, our aim is to find out whether light is necessary for photosynthesis or not. So what all we need? We need a potted plant. We need a green potted plant. We need iodine solution. We need scissors, one black paper, and stencil. So these are few requirements when we have to carry out this experiment to demonstrate that yes, light is very necessary for photosynthesis. Now pay attention to this solution, iodine solution. Iodine solution is a very important solution. It is a universal indicator of, can say universal indicator of starch. Universal indicator of starch. Wherever starch is present, if it will come in contact with iodine solution, there will be immediate reaction, and the reaction will be that part will turn uh, bluish black, and that indicates the presence of starch in any plant product. So we are using this iodine. What you have to remember, students, that iodine solution is a universal indicator of starch. It gives, you can also, we have tincture iodine at home also. We have potato at home. You can put one drop of, you will cut a potato, you will put one drop of tincture iodine on it, and you will see the reaction. If it will turn bluish black, this means potato has starch, and actually potato has starch. So this iodine solution, it acts as a universal indicator of starch. Now procedure. We have to take a potted plant and we have to de-starch it. Now this term de-starch its leaves by keeping it in dark place for 2 days. So what is the de-starching, children or viewers? We are keeping our plants in sunlight. They are doing photosynthesis. They are making glucose, and whatever glucose requirement that plant has, it will be utilized. Rest leftover will be converted into starch, and then it will be stored. So as and when the plant will need it, it will reconvert it into starch from glucose. It will be like this, children. Like we have glucose. Glucose it getting converted into starch, and when not required, if it's in excess. If excess of glucose is there, it will be converted into starch. If requirement will arise, then it will again convert into glucose. So what we are doing, we are de-starching, converting starch back into glucose. When we are taking a potted plant and we are keeping it in the dark area for 2 days, plant is not able to do photosynthesis and make glucose. What the plant will do, plant will utilize its that stored product starch. It will convert that into glucose. So this process is called de-starching. This is de-starching. So now imagine we have a potted plant. Earlier it was we have kept in our balcony, and now we are bringing it and keeping it in our storeroom for 2 days. Then what next step? We have to take a black paper and we have to cut a letter L. You can use S, you can use K as per your choice. But in this experiment, we have cut the stencil, this paper with letter L. Now what is next? Like this. This was the leaf. This was the black paper. So with the help of stencil, letter L was cut out from this black paper. And then this black paper, we have to cover the one leaf. Any leaf, it's still there in the potted plant. One leaf with black paper. So next is, now we have to shift our potted plant in which in one leaf we have done this experimental setup, and we have to kept this potted plant in sunlight for 4 to 5 hours. What will happen? Now the plant will again, it's there in the sunlight, will start doing photosynthesis, and all exposed part of the leaf, they will start making doing photosynthesis. They will start making glucose and converting it into starch. So now next step is, what we have to do, we have to find out that this area, the part of the leaf which we have covered with black paper, it has starch in it or not, because we have already de-starched the plant. Now all those parts of the plant which are exposed, they will have this starch in them, and not this one. So non-exposed part of the leaf, they are covered with non-exposed part of the leaf that was covered with black paper. It has not shown any color with iodine solution. Reason being, because there was no production of starch, and wherever starch was produced, it has shown bluish black color. So this indicates that if there is no light, there will be no formation of there will be no photosynthesis, there will be no formation of glucose, and subsequently there will be no question of formation of starch. So light is necessary. The part of leaf that was exposed to sunlight, including the shape letter L, it reacted with iodine solution and turned blue-black, whereas which was covered with black paper, no reaction was found in that. So no reaction of iodine with the covered part. The change in color in the uncovered part clearly shows that production of starch is there, presence of starch, and starch is a stored product of photosynthesis. Here, viewers, you have to remember starch is the stored product of photosynthesis. So here, starch was produced. Reaction is saying starch was produced. L later, which was we took out that part from the black paper. So starch, and this part is not showing any reaction. So there is no starch. So this means if light will fall on the plant, it will do photosynthesis and it will make glucose and finally the stored product starch. So with the help of this activity, we can prove that light is very necessary for photosynthesis. Next activity is also equally interesting activity and you will enjoy this activity. And you will feel, oh wow, we know that this availability of carbon dioxide is must and we can verify it with the help of some simple experimental setups. So here we are going to viewers, we are going to demonstrate with the help of one activity only, that is with the help of one activity. So viewer, next activity is we have to check that carbon dioxide is necessary for photosynthesis or not. If it's necessary, let's see if it's necessary, how we can verify it. So it's our second activity and our aim is to show that CO2 is necessary for photosynthesis. There are so many gases present in the air. Majority is nitrogen dioxide, then oxygen is there, argon is there, carbon dioxide is there, but this amount is 0.04%. But it's only carbon dioxide is necessary for photosynthesis. That is what we are going to demonstrate. So what all we need for this activity? We need a conical flask. This is the conical flask, viewers. Cork cap, one cap, just a lid. We need a cork cap. We need stand. There's a stand, potassium iodide crystals. We need iodine solution again because it's a universal indicator of starch. We need a potted plant. So this is our requirement if we want to prove that CO2 is necessary for photosynthesis. Let's have a look on the steps also. Take a plant with D-starch leaves. Now my viewers know what is the meaning of D-starch. So we have kept one plant inside our store for 2 days. It was not able to do photosynthesis. Whatever starch was present in that, it got converted back into glucose and plant was using it. So that is D-starch. So we are taking a D-starch plant, potted plant. What we are doing, students, we are going for one small, very interesting experimental setup. We are taking up the same conical flask, inserting one leaf when it's still there on the potted plant, and with the help of a stand, we are tying it to that potted plant. So now, interestingly, you must have noticed potassium hydride crystals. They are kept in the bottom of this conical flask, and then this lid is tightly closed with the help of cork. So, and this experimental setup means our potted plant in which we have kept one stand, one leaf is inserted inside the conical flask, and this conical flask is containing potassium iodide crystals. This is our experimental setup. So we will pick up the potted plant along with stand in this experimental setup, and we'll again keep it in the sun for 5 to 6 hours. So what will happen? This You must be thinking potassium iodide. Sorry, you must be thinking why potassium hydroxide? Conical flask, one leaf is inserted inside it, and crystals. So, viewers, please remember this question is there in NEET also. This question is asked in number of competitive exams, whether you are preparing for lectureship, even for 12th, even for CTET or your respective state uh, exams different. Even sometime it is asked in Olympiad, science Olympiad also this question was there. So why it is used? So this potassium, K is the symbol for potassium, O for oxygen, H for hydrogen. So this potassium hydroxide, it has the ability to absorb carbon dioxide gas. So this is the experimental setup. We will leave this experimental setup for 4-5 hours in the bright sun. What we will do then, after a few hours, we have to test that leaf which we have inserted in that conical flask, and we can also use some other for cross verification. So we will take up that leaf, we will put in boiling water. This can be carried out in the biolab only, but because we have to stay at home and we have to guide you about the basic procedure, so we are using this flow chart to tell you what all we do or you will do when the school will open, and you will perform this activity yourself in the biolab. We will take a beaker, we will put water in it, and we will we will pluck that leaf, the one which we have kept in the conical flask in this, and we'll boil it. After boiling, what we will do, we'll gently take this out and we will put it in a slightly bigger test tube which is containing ethanol. And again, we will keep this test tube containing the same leaf in the in the boiling water, and after a few minutes you will see that the leaf will start decoloring. It will start losing its green color. So ethanol is for removal of this chlorophyll, green color, so that we can go ahead with the testing without any interference. So when its decoloring is done, and but here, viewers, why ethanol is used? To decolor. It will remove all chlorophyll from this leaf. Second is, there is one precaution. Ethanol can catch fire very easily. That's why it is not, it is, this is boiling. You can see boiling water is kept in beaker, and then this test tube containing ethanol, it is kept there. It's a safety measure or safety tip. Then after 3 to 5 minutes, this test tube will be taken out very carefully from here, and the sleeve will be taken out and it will be washed gently. Thereafter, it will be kept in a petri dish. This is the petri dish, and we will take our universal indicator, that is iodine, and we'll pour a few drops of iodine on it. And then we will observe. So observation, what will be our observation? The leaf which was inserted in the conical flask shows no reaction with iodine solution. Surprising, na? No, it's not surprising, because you know that potassium hydroxide has the ability to absorb all the carbon dioxide. So whatever carbon dioxide was present in the air, which was there in the conical flask, potassium hydroxide has absorbed it, and that's why the leaf was not able to do photosynthesis, and there was no change in color. So conclusion is that reason is potassium hydroxide, it has absorbed all carbon dioxide. That's why that leaf has not shown any color, any change in color when we poured iodine, drops of iodine on that leaf. So with the help of this experiment, viewers, we can say that yes, carbon dioxide is very necessary for photosynthesis. And I'm happy that my viewers, if you have gone through this video completely and you have gone through this activity completely, you will retain it and you will remember it. What you will remember, that iodine is a universal indicator of starch. Second thing you will remember, that potassium hydroxide has the ability to absorb carbon dioxide from the air. And third thing is that because of this potassium hydroxide crystals, there was this potassium hydroxide has actually eaten away all the carbon dioxide which was present in that conical flask. So no carbon dioxide was present and there was no photosynthesis, no raw material, no photosynthesis. Sunlight was also there. This plant was potted plant in which water supply was intact. Then you can say the green part was also there, but carbon dioxide was missing. And that's why there was no photosynthesis and no formation of glucose and no formation of starch. Now, viewers, you must have come across beautiful leaves with variegated leaves. Leaves. Yes, viewers. And next is a equally interesting segment of this video, and here you will come to know about the photosynthesis ability of variegated leaf. Let me tell you what is variegated leaf. The leaf in which is not completely green. It has patches of blue color, yellow color, maroon color, violet color, purple color, and different pattern, different designs. So those plants, such leaves are called variegated leaves. Now, till this, you know that for photosynthesis, chlorophyll is must. And but you must be thinking that if the leaf is variegated, like croton, they have variegated leaves. Coleus, again, variegated leaf. Some part is green, some part is violet, some part is brownish, maroonish. So how they are doing it? How they are doing photosynthesis? The entire leaf is doing photosynthesis. Only the green part is doing photosynthesis. So we have to explore this in now. So for that, let's together see this also. It's very interesting that green part, majority of the green part, it contains chlorophyll. And if it's a part of the leaf, it's colored. It has some other pigments. In you are in class 7, so you can only remember that xanthophyll, carotene, carotenoids. A few these are complicated terms for you, but they are present and they are giving color to the different colors to the leaf. But chlorophyll is present in only green part. Chlorophyll, a few pigments of chlorophyll can also be present in the variegated part, the colorful part, but the amount is very less. So let's see if such leaf is used to demonstrate which part is doing photosynthesis and which is not, what will be the observation. And for that, I'll take you to the next activity. It's at presence of chlorophyll. That is must. It's our third activity. So presence of chlorophyll is must for photosynthesis. So for that, what we have to do, again, we need one variegated leaf. Okay. And I want to show you a few variegated leaves also. Mentioned about croton and coleus, but we have many other, like I'm holding Dracaena. If you will see this leaf closely, let me show you closely. See, this is leaf of Dracaena. And it has the central part is green in color, whereas the margins, they are creamish. So this is again a variegated leaf. So which part is doing photosynthesis? You know, chlorophyll is necessary for photosynthesis. So till this, we will say, or my viewers will say, ma'am, this green part, it is containing chlorophyll. So it will show reaction. Explore ourselves with the help of one experimental setup, that whether the chlorophyll is present in only green part or in the non-green part also, and which part of the variegated leaf it does photosynthesis. Very interesting. So let's explore it together. So viewers, now we are going to explore one very interesting thing, that which part of the leaf, green, non-green, both, or only green, is necessary for photosynthesis. And our aim is that to verify chlorophyll is necessary for photosynthesis. So for that, we need one variegated leaf. It can be Dracaena, it can be Coleus, it can be Croton, or even it can be money plant. You must have seen in money plant also we have a variation. Some patches or bluish or whitish part is there. So we need iodine solution. Now you know this says universal indicator Iodine is universal indicator of starch. We need ethanol because ethanol, it dissolves chlorophyll content and helps in avoiding any kind of interference when we are checking the leaf for the presence of starch. I'm talking about the reaction of starch with iodine. And we need water, watch glass, beaker, burner, the same experimental setup actually which we have used for the previous activity, where we have proved that CO2 is necessary for photosynthesis. So what we will do, we will pluck any variegated leaf. It can be money plant, it can be any, and which is exposed to sun for a few hours. And thereafter what we will do, we will draw its outline on the paper, and we will also make that which part was green, which was non-green or variegated. We'll draw which after we have to test that leaf for starch. The procedure will remain the same: boiling in water, then boiling in ethanol, washing in water gently, spreading, keeping it gently on the watch glass, putting iodine solution or pouring iodine solution on the entire leaf, and then observing further reaction. So if we have taken money plant, this was initially when literally it was plucked from the plant, and then after reaction, you must be noticing that all yellowish part, they are still yellow. They have not shown any color with iodine. They are still yellow. Whereas all green parts of this variegated leaf of money plant, they are, after experiment, they have shown bluish black color. So this means this green part is, or the chlorophyll containing part, is actually responsible for photosynthesis and is actually necessary for photosynthesis. So no change in color with iodine solution in white or creamish part. It clearly indicates that there is absence of chlorophyll, or the pigment is present in very least amount. No reaction. Whereas the green part, it has shown reaction with iodine. This means it is necessary for photosynthesis. Photosynthesis has obviously taken place there, formation of glucose there, and the end product, it was further converted into stored product, that is starch, which has given this reaction with iodine solution. So with this activity, we can prove that chlorophyll is necessary for photosynthesis. So today again, it was a interesting session where you have not only learned about the conditions for photosynthesis, but we have also tried to know about those conditions, whether they are actually necessary or not, with the help of certain activities. I want my viewers to just give me their feedback, whether they have liked this video or not, or whether this video was of some help to them. If it was some help to them, I request all my viewers to uh, subscribe my channel for so similar kind of more videos. And if they think that some improvement is there, please do drop your comments. They will be valuable for me, and they will help me to make this content little more viewer friendly. So the learning outcome of today's video is we learned about conditions of photosynthesis, and we have also seen how we can experimentally prove that actually they are. Light is necessary, carbon dioxide is necessary, water is necessary, chlorophyll is necessary. So that's it, viewers. So no video is complete without a worksheet. So here also I'm sharing one page in which there are 5 questions, and very easy question. I request you to either do these orally or in written form, whatever. And next time, if you will surf online my biohub, you will come across my next video on this chapter, that's chapter 1, nutrition in plants, from class 7. I'm going to take up part 4 of this chapter in my next video. So stay tuned with me on my YouTube channel. It's Biohub, and I'm your facilitator, Dr. Seema Sharma. Till then, take care. I want you all to I want to tell you, viewers, whether you are a student, a parent, or a teacher, that this world is closed for renovation and there will be grand opening soon. Till then, we all have to follow the COVID protocols. We have to stay home and only then we are safe. So hope to see you all again with my next video. Till then, take care and it's bye bye from your facilitator. Bye bye.